genetics. This video and modular system was produced by the University of Wyoming in Bray. We're going to dive into what Ipyrad does to process our RADseq data and assembly. So to process RAD or DD data efficiently, especially in our cloud-based environments, we're going to use this pipeline called Ipyrad. You can read more about it at their website if you would like. It is a modular, scalable, and well suited to high throughput data with built-in support for RADseq and DD RAD data types. Ipyrad works in 7 main steps, each of which can be run independently. We're going to walk through these steps just to see how the system handles our raw data all the way to our analysis-ready assembled data set. So step 1 is all about organizing our data. If we have raw reads from pooled samples, Ipyrad will demultiplex them using barcode information, assigning each read to the correct individual. If our data is already demultiplexed, Ipyrad will simply load the FASTQ files into the analysis, and this forms the basis of a named assembly in which we will define, which defines the full workflow. So that's the step 1 in sorting. The second step is to filter or edit our reads. So next, Ipyrad filters the raw reads based on quality scores. Low quality base calls are masked as ends, and reads with too many ends are discarded. We can also trim adapters and clean up restriction cut sites, which is especially important for RAD-DD reads where edge quality tends to be low. In the third step, our clustering step, we group similar reads within individuals. This is either done through de novo clustering or by mapping to a reference genome. The goal here is to find and align stacks of reads that likely represent the same locus in each sample. This sets up for accurate consensus sequence calling later. In step 4, we're going to in step 4 and 5, we're going to create our consensus sequence, which includes estimating heterozygosity and error rate in step 4 from our clustered reads. And this step is critical and helps Ipyrad make more intelligent base calls later on. Within step 5, our consensus base calling step is where we will get our consensus sequences for each locus per sample. Ipyrad uses a binomial model to decide what the base at each site should be based on the read depth, error rate, and heterozygosity. So at this point, we will also filter out loci with too many ambiguous bases within this step. Step 6 is clustering among all samples. So now that we have consensus sequences per sample, we will cluster them across all individuals to find the homologous loci. Again, this step can either be done de novo or using a reference, and the sequences are aligned to produce site-by-site comparisons across the data set. And then finally, in our final filtering and output step, Ipyrad applies final filters, such as setting minimal number of samples required per locus, and formats that output into files for downstream analysis. These include VCFs, Nexus files, and Ipyrad-specific formats that are ready for population genetics or phylogenetic analyses. There are a couple things specific to the submodule 1 that we'll go over. So within this submodule 1, it runs within the Jupyter notebook, but within a Python kernel. So as you're going through these things, you should see an exclamation mark for cells that use, or for cells with a single line, or 2 percentage signs with bash after it, which will run a whole block of code. However, this is only seen in module 3. And this allows us to run the bash code from a Python kernel. And then finally, at the end, you should get an Ipyrad out folder that lists all of those specific files that we were taught or that I just mentioned. And finally, you will take this Ipyrad output and then start to put it through some basic population structure analyses in R that include some basic statistics, principal component analyses, SNMF, and isolation by distance, or IBD. Again, these run in Jupyter, but in an R kernel. So we're running these in Jupyter, but in submodule 2, they are within R.